This Week in Virology, the podcast about viruses, the kind that make you sick.
Hi, I'm Rich Condit from Microbe TV, and I'm here in Chicago at the International Hepatitis B virus meeting, abbreviated HBV. And I'm here with Stephan Urban, who is a professor at the University of Heidelberg.
That's correct.
And also an HBV researcher. And we're going to talk about his life and work for the record. So, Stephan, welcome.
Yeah, welcome. Thank you for having me.
I'm glad we finally get a little time together. We were actually at a restaurant last night that was very noisy, and we tried to have a conversation, but we had a good meal. All right.
We had a good meal. That's true, yeah.
So I'm interested in your history, like the whole thing. Where were you born? How did you, your career path, and how you got into hepatitis B virus?
Okay, yeah, let me start from the beginning. I think one of the, let's say, first thing that was very interesting for me in school was chemistry. So I started as a chemist. I studied chemistry and biochemistry. And even before leaving school, I had a deep interest. So I was having my own laboratory in the cellar of our house, and did some experiments that you should not tell openly what we've done. Had to do. And actually, it became serious. So I was starting to do really organic chemistry, and I participated in a contest, a German contest. And when I was 18, I won the first prize in German contest.
Did I see a picture of that?
Yes.
There's
a stamp. There's a picture
great.
when I was young and did that stuff. And that then led me finally to study chemistry. But I was mainly interested in biochemistry. That was not taught in school, but at the university it was then. I went to Tübingen, and there were three, at that time, 1980, 82, that time, only three places where you can study biochemistry as a separate study, in Berlin and in Tübingen, and I had chosen Tübingen. There's a long tradition, Buchanan, Nobel Prize winners, and so on, pheromones, and I liked it very much. And I also studied then chemistry, biochemistry, and philosophy a little bit. I was always interested in a little bit more than just the science. I always wanted to know what science means for society and so on. And there was one program in that study that was you had the choice to go as a student into the Max Planck Institute for Biochemistry in Munich. That was at the time when the Nobel Prize winner of Robert Huber was there, and I was visiting several research groups. So I was directly involved in research as a practical student. And that's the first time I came in contact with hepatitis B virus. So I was not looking for virology. I was just realizing, oh, wow, this is a beautiful thing to learn via viruses about the hosts and about the cell and about all these mechanisms, many of these principal or fundamental investigations that have been found for cell biology have been found using viruses. And it was about the time that both Peter Hans Hofschneider was one of the big ones, interested as a medical doctor in carcinogenesis, very far away from replication, but I liked it very much, and I decided I had to go back to Tübingen, do my diploma at that time, and then I could decide to go for a PhD study to go there. And at that time, I studied already hepatitis B virus, so it's more than 30 years ago.
Wow, okay. So all the way from what we would call an undergraduate, right?
Yeah,
okay,
yeah.
All right, excellent.
And actually, this was more related to medical questions, and I was more interested in the virus at that time. And I have to decide to go for a postdoc. And of course, normally you then go to the US, and I was quite on the way to the US, but private reasons, or girlfriend, then came in between. And
life got in the way, yeah.
Exactly. And there was another big guy in Germany. This was in Heidelberg. That was Heinz Schaller. These are my two mentors, Peter Hans Hofschneider, Heinz Schaller. And he headed the hepatitis group at the Center for Molecular Biology, ZMBH, in Heidelberg, which is a very famous, still is, very famous center there. And he was looking for a last postdoc, I think, and I went there, and many applicants. And for some reason, he chose me, and then I did my habilitation. But I switched the field. So his view was, we study viruses because we want to know about viruses and not so much about hosts and so on. And he wanted to understand how that virus replicates. And as you might have heard from that meeting here, it's a very special virus. Yeah, so it's a small virus, very compressed genomes. Regulation is on a minimum of genetic elements that regulate it. A lot of interaction, kind of zip virus. Yeah, you have a lot of information in very few overlapping genes and so on. That fascinated me, and the receptor was not identified at that time. The point is, a lot of interesting observations were already out. For one example, you can use 2 or 3 virions, particles, to infect a chimpanzee. How does that virus find the liver? And it's exclusively in the liver. But we could not study the virus in cell culture, so there was no cell cultures or even hepatoma cells, so they were susceptible. And the question, what is the receptor, was a hallmark. We needed to know that in order to, for example, produce cell culture systems by transfection, etc. And so I started out with that. And the system that we had that was interesting was that after discovery of the HBV virus, many other or some other viruses at that time were found. For example, the duck hepatitis B virus in Peking ducks. Yeah. There was a woodchuck hepatitis B virus in woodchucks, but woodchuck are not a very suitable animal system, as you can imagine, quite aggressive and so, but still people have done work on that, especially because they develop cancer. So the hepatitis B virus is a carcinogen. It's really, worldwide, I think it's just behind cigarette smoking. Is hepatitis B virus. And we use that duck system. So the first point was we need to have a flock of ducks that are not already infected, because quite many of these Peking ducks were infected at that time.
So the ducks are infected in the wild?
Yeah. Yeah, you find them. And if you go to a lake or something and you see some ducklings, you take a PCR out, you will, of course, amplify duck or even other avian hepatinoviruses.
Okay.
So we had to find some that were not infected yet. We put them in a village close to Heidelberg, and we had a flock there. Yeah, and occasionally we went there, and these small ducklings. Yeah, then we took one, and then we prepared primary hepatocytes. Yeah. And we infected them. That would be very nice. So I took a lot of time in order to trying to identify the duck hepatitis B virus receptor in order to find out maybe the homologous protein in humans. Took quite a while. We found the receptor, we and another group.
At this point, you're still a postdoc, right?
I was a postdoc, yeah. Okay. And I was on the way of habilitation as we have it in Germany. Right. Yeah. So in order to make, for me, it was very clear to say from the beginning, I wanted to stay in academics.
Right.
That was clear.
You're already on the path. Doesn't matter whether you're a postdoc.
Doesn't matter whether
you're on
I'm
the
a postdoc
path.
or employed.
Okay.
I wanted to go for I was quite sure about that, yeah. And we found that receptor and we characterized the receptor, and that was the basis for the habilitation. And the approach, and that was important, the approach to do that was that we studied fragments of the envelope protein of the duck hepatitis B virus. We synthesized them in E. coli, and we synthesized them by chemical synthesis, peptides, and there were modification, and we made inhibitory tests. So we looked whether these fragments could interfere with infection of the primary hepatocytes. And
so at this point, you've got primary hepatocytes in culture, right?
In culture, yeah, but of course you have to prepare them all the time. Yeah, so we occasionally every other week we were.
And can you grow up enough virus on these so that you can routinely do infections?
Yes, we can grow the virus.
Because I know that growing in a lot of systems, growing the virus is difficult.
No,
but this
that was fine. That worked,
Okay.
and that was the reason we've chosen them. Of course, people tried to do the same in human hepatocytes.
Right.
It worked to some extent, but it worked not on a regular basis yet. We did that later on then.
Okay.
But first of all, we had the determinants in the envelope protein that are important for receptor interaction, and with that approaches, we could then identify the receptor, we and others, and characterize the receptor. It was carboxypeptidase, and and then I spent another 2 or 3 years in order to clone the human receptor. I mean, at that time it was not so easy just to order a clone on the gene bank somewhere at a company. And it was very clear it is not the human receptor, so it was a little bit frustrating. Yeah, but what we had.
So you identified the dark receptor, cloned the same gene from humans, and it's not the receptor.
That's not the receptor.
Okay.
No.
Good.
No, not the human homolog.
Right.
For sure.
That's a standard frustrating story.
It's
Good.
a standard frustrating story, but not the most frustrating stuff yet to come. But the important point was we had peptides and we had an assay how we can do that in humans. And about that time, primary human hepatocytes were available or occasionally available, mostly by resurrection of parts of the liver from cancers, and you could, with an ethical approval, take some of the hepatocyte aside and then test that. And the first cell line in 2002 came out that could be differentiated, and it's a progenitor cell to a hepatocyte that's susceptible. And with this, we could then do these experiments. And we repeated the approach in human, with the human hepatitis B virus. And there was a collaboration with a French colleague of mine in Rennes. So I have to go to Rennes, 900 kilometers all the time, to do an experiment.
Are you still a postdoc?
No.
Now you're a professor.
No, I'm a professor.
Okay.
So at that time I was a postdoc. Yeah. And then we try to determine these regions that are important in the human envelope protein, which is quite different. It's from the structure, from the general structure, it's similar, but from the sequence and probably the target is different. Yeah, and we were sitting there in the evening having French wine and discussing all the night, and then we designed new peptides. I went back to Heidelberg, made these peptides. Yeah. And a friend of my former boss who did not, he thought, there's also a story because, peptides are expensive, so I need some support. And there were some professors at that institute that gave me some of these peptides from their budget, yeah. Not my boss. He said you should make that recombinantly, etc. Yeah? And we designed that, and we made an experiment. I think it was summer Christmas, and put them on the cells and infected them. And then some week later, Philippe, that was his name, called and said, they are extremely efficient. So we had nanomolar concentrations. We thought we made a mistake, yeah, but we saw these peptides are extremely potent in inhibiting HBV infection. Then we made mutagenesis and we found out, okay, these are the ligands for the unknown receptor on that hepatocytes. And then, of course, we tried to make cross-linking experiment and everything like that for years without any success. But we defined the peptides, we defined amino acids within, and we came along with the idea, can we use them as a therapeutic? Can we use them as an entry inhibitor? Yeah. And about that time, there came one peptide, maybe you know that T20 and fofetide for HIV. So Roche developed an entry inhibitor, first entry inhibitor as a peptidic entry inhibitor for HIV. Problems with resistance and efficacy, you need 180 milligram per day per patient. But they showed you can use peptides and produce them in tons that was a pro for our development. On the other hand, peptides, they are not very stable, they're degraded, etc. And thinking, do we go for peptidomimetics or will we really follow and optimize these peptides? And with these new systems that we had, although it took a long time, every experiment 4 weeks, we really made hundreds of peptides and mutated every amino acid to look what is the determinants. And we started preclinical development and shown it in vivo. These were experiments done in mice that have a deficient immune system and were transplanted with human hepatocytes, a very complicated system. But in those mice, we had very low, 0.2 milligram per kilogram were sufficient to completely block infection. I would say that was a kind of breakthrough. There was a Nature Biotech paper that was for me also a kind of breakthrough. And the basis of that, then we thought, let's go for clinical trials. Let's go for the production. Learning by doing, I was not used and nobody taught me at that time how to make GMP production of peptides, how to perform toxicity studies, pharmacokinetic studies, protocols that can be used for application for the authorities, for example. But we went through that process. And we called that peptide Mercludex. It was a funny name. A postdoc in my lab was thinking about it for exclude with a muricylated peptide hepatitis. Yeah?
Okay.
Mercludex. And some people still call it Mercludex here. Yeah. And then we started really phase 1 clinical trials. And
phase 1 clinical trial, you're just testing to make sure that it's not toxic. Is that right?
Yes, exactly.
You're not really testing for efficacy.
No, there was no efficacy, and we got, that was the important point. We had a small company who was who had interest in the license and licensed this in, but they did not have the monetary background to really put in millions of dollars or euros.
At this point, let's put a date on this. What year would this be for the phase 1 clinical trials?
That was exactly 2 days after my 50th birthday at the 27th of July in 2011.
Okay, now we got your age. This is good.
Now you can calculate. Exactly.
All right.
And of course, that was really great to have phase 1. And of course, we tried to get big pharmaceutical companies interested in advance, because all the preclinical data in the mouse system and the cell culture system were really convincing because of the activity. Picomolar concentrations were sufficient to block infection. No expectation of resistance because you have a target, but we didn't know the target when we started the clinical trials.
And at this point, is this just a raw peptide?
Yes, it was a raw peptide, but also, of course, I have to make a formulation. Yeah.
Okay.
And I was thinking, I mean, it was very clear we could not orally apply the peptide. That was clear. So, but we had a formulation for subcutaneous application.
Okay.
And we tested that, of course, in several animals. So it has to be tested in cynomolgus monkeys and in dogs and in rats and so on. That was all done at the university, mostly at the university, some contractors that can do it according to the protocols.
And the phase 1 probably has dose studies in it, so you go up to something well beyond what you think would be physiological.
So we pushed more and more, and we could see the pharmacokinetics. Interesting, because within minutes it ended up in the liver. So these peptides represented these exclusive hepatotropism that the virus have. So that make us quite confident.
Very interesting, yeah.
And one amino acid exchange, an L amino acid to a D amino acid, completely abrogated activity and pharmacokinetics.
So this has a real high affinity for something that's in the liver.
Yeah, but it was not known. And that, of course, made these first clinical trials also more complicated because you have an unknown target and you have to go slowly with the dosing, but we were up to 20 milligram and more per it was not a patient at that time yet. Yeah, it was a volunteer. Yeah, and no toxicity, no side effects. And then, of course, the big question comes: is there efficacy? And the point was,
let me back up for just a minute here, because I just want to make it clear for some of the listeners that this is important for the toxicity studies, because clearly you're looking at a cellular target. You don't know what the target is.
Yes.
It could be important,
Yes.
right?
Yeah, absolutely.
So this could be toxic, crazy
Absolutely.
toxic.
Absolutely.
Okay, but
and
it
there was just a year before a clinical trial in London, where they tested an antibody in a phase 1, and it was absolutely toxic. It was a disaster, And then the regulations were quite stronger. So, for example, we couldn't inject in 3 people in parallel. We have to make the low dose in series, and so you have to do that, and the committee decides when you go to the next concentration and so on. And so we saw no toxicity or nothing that was concerning us. I mean, the people in that situation, of course, are concerned. They are isolated and in a very close clinical environment with an intensive care station just around the corner in case something happens, and they are there for a week, So this is, of course, something, headache or something happens, with this. And then something happened that was really very important, and that was first the identification of the receptor, which was as expected, a liver-specific transporter, which is just only expressed at hepatocytes. This was when Huili's work, who was honored in that meeting. We also were looking for that. We also confirmed that this is the molecule. And with that as a target, we knew where we had to look for possible side effects, natural changes of the natural function of that transporter, which would be bile acid level, would be elevated. We, of course, immediately implemented this kind of thing.
So, pardon me, I'm clueless about, liver function, but a bile acid transporter, is this going to move bile acids made in hepatocytes out into the bloodstream? Is that what its job is?
It's actually the vice versa, but on the basis of the following, because the liver makes the bile acids, and the bile acids go through the bile duct into the intestine, and they help to solubilize. And then they are reabsorbed in the gut and back transported to the liver.
And that's what this transporter is.
And that's where the transporter acts, because it brings it back into the hepatocyte, a kind of reusage of these kind of bile acids. There are several of these transporters. It's also important. And the one called NTCP, this transporter is a main bile acid transporter, but selective for some conjugated bile salts. So this was, on the one hand side, good because we knew the target. On the other hand side, elevated bile salts were always associated with itching and with side effects at that time. But one didn't know whether this specific bile acid transporter is the one that then is the causative transporter for this.
But now you can look at serum bile acids.
Yes, of course, we do that regularly. But there was another observation that was, there was a polymorphism discovered by chance in a pregnant woman, and the polymorphism was in that very molecule, and this woman did not have any symptoms. The doctors just came to the idea, this elevated bile acid, to check for the sequence of the transporter, and found that the NTCP is knocked out in that woman, homozygote
knocked out.
Yes.
Okay.
And then came out that there are many people that have this polymorphism, either heterogeneous or homogeneous. They don't show any symptoms. They live without a bile acid transporter, and they are not very susceptible. It was, of course, then, like in HIV, it is a very similar thing. That some essential receptor were. I think that was something that made us quite confident that we can use that cellular target. Another thing that was very important was that we didn't need to, and still with the now approved drug, don't need to completely saturate the transporter. It is still active against the virus. The hypothesis is that you probably need a trimer for the virus entry. And if you just block one of these three domains, you abrogate the function of the receptor, but you still keep some of these transporters functioning. And then, of course, the very important point came that we have to enter into a phase 2 clinical trial. And the whole thing at that time was first, I have to add that, we haven't even mentioned it yet, was going for HBV, hepatitis B virus. And there was a big concern or a big, let's say, skepticism that an entry inhibitor in a chronic infection of a virus that persists via cccDNA on a long-term run makes anything in a chronic setting. Of course, we first had the idea, okay, let's prevent mother-to-child transmission, for example. Let's prevent reinfection of the liver in after transplantation. That was a problem. Yeah, people with hepatitis B needed transplant, and you have to prevent the reinfection of the liver. Yeah, we do that with antibodies, but that would work. Yeah, or needle stick injuries. Yeah, post-exposure prophylaxis. That was the thing. But what would happen? And that was the concept that nobody believed in. And that I thought still at that time, and still think so now, is important. What happens if you constantly block de novo infection of hepatocytes during the regeneration of the liver? You close the door for de novo cccDNA formation, and let the liver regenerate and look whether you get more and more and more. Of course, it was clear that it will be a long-term therapy. It will not directly affect the virus. Because it's not directly interfering with replication. What would happen if you do that? Would you lose at the end maybe the virus? Yeah? This is the one point. The other point was, of course, going through, nobody at that time, by the way, were interested in HBV cure. We had good drugs already, nucleoside analogs, that suppress virus replication. They are not curative. You have to take them more or less lifelong. Yeah. They develop better and better. Several of them, we mentioned them already, are now regularly used, entecavir or tenofovir or derivative. They are cheap, they are affordable now, they go into these countries where you need them. They are good, but they don't cure. And the problems you wanted to cure in principle, and you wanted also to address different pathways that are important. That was my idea. But at that time, the pharmaceutical companies were more interested in hepatitis C because that problem was not solved yet. Right. Yeah, that the boost for hepatitis B came later. So we were quite a little bit earlier with our development, and therefore the interest was not very big. And what we did not mention yet is something that's the route that we went clinically, and that's the hepatitis delta virus that comes now into play. And the hepatitis delta virus, maybe I explained it a little bit, is a parasite on a parasite, we can imagine. It is a RNA, the smallest RNA virus that replicates in the liver, that was described in 1977. But how does it come to the liver? So the interesting thing is the virus evolved a way to hook up to the envelope proteins of the hepatitis B virus, which are self-assembly competent. So it just needs a hook, which is at the very C-terminus of its only antigen, the delta antigen, and then it is enveloped in hepatitis B virus. So from outside, it looks like a hepatitis B virus, but once it entered the liver, it replicates by a completely different mechanism.
At the same time, it's completely dependent on hepatitis B for its replication because it needs that protein, right?
But in principle, it doesn't need the whole replication of hepatitis B, just needs the envelope protein. Okay. Yeah? So, but that, of course, was also interesting to look whether our drug works against hepatitis delta virus. It should, and it did. So with the same efficacy, we can block entry of that hepatitis delta virus, which co-infects, of course, as you may realize, only those patients or only hepatitis B virus infected patients. So we have a kind of super infection of Delta virus on the chronic HBV patients, and we guess it's about in the range at least 12 million or maybe even more. So 25 million people are co-infected with both viruses. About 5% of all B virus-infected patients are co-infected with Delta, and that is in hotspots. We don't in some regions, Mongolia, for example, or Pacific Islands in Brazil, you have very high prevalence of Delta virus as co-infected. In other regions, that's different. And we don't know because we don't test. But that's just aside.
Do you know why there are hot spots? This is an aside as well, but do you know why there are hot spots? Or
I can tell you one interesting story that we made. We are developing also a point of care test for Delta now, because diagnostic is important. And we had a collaboration in a small village in China, where it was known that a lot of HBV in China, HBV is very high, so the prevalence there. And in that region, people used to inject drugs for social reasons. So caffeine drugs, so not hard drugs. So they inject caffeine. And it was known before because we had a collaboration with a professor there, and he showed us, he showed that there is a very high prevalence of hepatitis C virus, obviously transmitted. Same sort of transmission, And this is probably the transmission that is intravenous, so intravenous drug users have it preferentially. Sometimes it may be, like in Egypt, through very close, so iatrogenic by some medications that have not been tested. Probably when you live in a high prevalence region, in a family where the several members are infected and someone brings in Delta, and then I think by sharing things, you can propagate that. Mm-hmm. And once you overcome a specific threshold, then probably you will have higher thing. But we don't know exactly why that is. We don't know exactly whether there is, why there are some regions where it's completely obviously under control, in other regions where you have. Africa is probably huge. We have done some studies in Africa, but also there in some rural cities, rural areas, we found high prevalence. In some cities we didn't find. We're just lacking good data for that. Yeah. But to come back to the biology, for us, for Delta, there was no drug approved. Yeah. Right. And these people did just not respond to the HBV drugs. So the HBV repression of with nucleoside analog did not help to lower the Delta virus, because the envelope proteins of the B virus are not affected by the nukes. Yeah. And
we should emphasize that the Delta superinfection over HBV greatly exacerbates the disease.
Yes, correct. Absolutely. All right. So absolutely. These people accelerates everything. Right. Yeah. So carcinogenesis accelerates, cirrhosis is accelerated, and so we were thinking on the basis that we could show, it works for Delta. What happens if we give that in Delta patients? So we made a sub-study in one of the B studies and looked, and it was a very small study at the very beginning. I think it was only 24 patients, but it was immediately visible in these few patients that it has a profound effect if you treat for 24 weeks. That was the longest time we could treat at the time. The people normalize ALT level by a monotherapy. They lower their viral load, and then the idea was, let's follow for the Delta, because there is the highest unmet medical need, larger phase 2 studies were then designed just to look in Delta patients, which was not so easy because were not so many patients already identified. So we had to go to clinical centers that have Delta patients, occasionally large clinical centers. There were clinicians in Hanover, for example, in Germany, some in Heidelberg as well. And that was then very, very successful. And the people really profited from that study. And in Europe, there is a program, and then we received something that's called prime eligibility and orphan drug designation. Orphan drug designation from the FDA and from the EMA, the European Medical Agency. And there is this program. If you show efficacy in phase 2 studies, and you have already planned a phase 3 study, and you provide the agency continuously with data from the phase 3 study, you can get a conditional approval. So they wanted to make faster access to those patients. There were also national programs, for example, in France, it's called the ATU program, where even before approval, conditional approval by the European Medical Agency, they allow this drug to be used in these clinical centers. And that then happened. And we got conditional approval of that drug in 2020. Yeah. And that was then really, I would say, a breakthrough, because from an academic and a small company, which of course also recruited venture capital, but not in that dimensions as you normally think about drug development, we could come to that point. And then big pharmaceutical companies became interested.
Before I just want to back up for 1 minute. So this is still being used as the native peptide? Is it modified at all?
Of course, it is not. It is not the carbon copy of the peptide in a natural virus. We have optimized it. It has been shortened, optimized for the size. We have done some some amino acid exchanges because of solubility, these kind of things. But it is the peptide. It's really
and it's still administered subcutaneously.
It's subcutaneous. That is a drawback, I would say. But we did some
but
work.
You're talking about a nasty disease, so.
Yeah, and what we also realized, we were fearing compliance of patients, but in all these clinical studies, we found obviously people accept that. They go once a day to their refrigerator, and once they realize that this really helps them, they do it. So they stay with that one-day administration. Of course, would be better to have a slow-release formulation once a week, but it's not a principal hurdle. Of course, it would be better to have a pill a day, but they do that, and that works.
So this is effective against hepatitis Delta.
Yeah.
And I want to make sure. How about hepatitis B? Do you know?
In all models, it works very efficiently in preventing hepatitis B virus infection. It is as efficient for de novo infection of hepatocytes for hepatitis B. It will probably not, and we had one study with B monotherapy, and it shows some virological effects, but it's probably less efficient, or you need longer treatment durations until you may eliminate the CCC DNA, because there are different mechanisms of persistence that are still working. Nevertheless, I think the important thing, as we later then learned, is also combination. If you use bulevirtide now, and now I really refer to the latest clinical trials that then were designed for a long time, because, as I said at the beginning, we need to wait for a long time until we get elimination of that virus. So there have been trials performed, 3 years treatment and then 2 years follow-up. They are just finished, so you see it's long term design, that the monotherapy alone is very good. So you normalize ALT levels. Many of these patients have negative virological markers in the serum. But if you remove the drug, you still have residual virus, and it rebounds and can re-amplify as long as the HBV is not cured. And that poses, of course, a problem, because you have to continuously give it as a monotherapy. But if you add it now, or if you combine it with interferon, and the reason, let me say the reason, we are of course working further. We're not only doing drug development, we will try to understand what's happening. If the Delta virus can enter an hepatocyte, and this very hepatocyte divides, then the Delta virus can propagate via cell division.
Okay.
So that means it has a second way of persisting in the liver, not only by the external pathway, using the envelope proteins of the B virus, but also by a cell division-mediated spread, as we call it. And interestingly, interferon, which was used as a therapeutic approach for Delta, although it was never approved. Interferon is not approved for Delta, but doctors used it because they had no alternative. If people can accept interferon therapy, many of these people are so ill they cannot, they are not eligible anymore to interferon. But if you do that, with interferon you can prevent the cell division mediated spread. And if you hit with both, and we have done that now, or Gilead, who bought the drug, has done that now, then you really have the chance to up to 46%, let's say cure, or at least they have a sustained virological response 2 years after follow-up. That means this hitting it with these both ways of the pathway, you may really get a curative treatment for Delta, although they are not cured from HBV yet.
Right.
Yeah, so we have something that was unexpected. That means we can eliminate the virus without eliminating the helper virus. Of course, it would be best to eliminate both here, but we are not yet done at that stage. Yeah.
So still work to do.
Still work to do, yeah. And of course, we're doing these kind of things. Yeah, we're looking for direct replication inhibitors and try to understand what happens in patients, responses, and so on. Yeah.
Fascinating story. I have one additional thing, if you don't mind.
Yeah.
If I think I noticed the Stephan Urban Foundation.
Yes.
What's that?
Okay. Let's talk about money. That's okay. So, I was normally, it was not I did not do science for making money.
Right.
But the consequence of the whole thing was that the company that had the rights was bought finally by Gilead. And that was quite a big deal. I don't want to say the numbers, but it was quite a big deal.
Quite a big deal. It's good enough.
And of course, I personally profited because I was involved as an inventor and patent rights and so on. So I had personal profit from that. And my two advisors, or as I say, Peter-Hans Hofschneider and Heinz Schaller, one of the things they were convinced of, if you make this occasional lucky situation, if you make money with science, give it back to science.
Excellent.
And for me, the important, this is a Stephan Urban Foundation, so I put not all, but quite some of this money into this foundation. And I realize more and more how important that is to be efficient with that money. I don't want to support projects that can be supported or find money by natural governmental grants giving. But we have to invest into public health issues. We have to bring now these drugs, and that will take a while, bring these drugs to people in those countries that do not have a lot of money. So, for example, some of the grant, and we have to bring education in those countries. Yeah, Africa or in some Asian countries or thing, and that is the purpose of that stuff, to bring money into some kind of fancy ideas, maybe. Yeah, also be trying to then fund some projects. It's not yet so far. At the moment, we just support people, so scientists that want to come to these conferences or projects in Africa, or diagnostic projects in Africa, because we need to diagnose the patients, and this is the idea on that foundation. Yeah.
Outstanding. Stephan, this has been, what a great story.
Thank you.
What a nice story. Congratulations to you, and good luck going forward.
Thank you very much, Rich.
And thanks for talking with us.
Thank you for inviting me. Thank you.